Historically, our company essentially was not that hybrid, and that our computational experimental platform was still in its infancy, and we started with a stronger computational focus. Most of our work was done, our validation work was done through experimentalists at our partner pharmaceutical companies or through contract research organizations. And over time, we were able to evolve and make decisions regarding our own internal lab build, what's cost effective to bring internally, and what tools and stuff are unique to our company. That's pretty protein cure specific. So I was trying to help when I said about lessons for this lightning talk, it was a little bit about thinking on a bigger level about what there are some non-technical or non-protein cure specific principles that I think are pretty helpful to think about. So I'm going to call these the search over the 7 C's. It's an acrostic where I have 7 lessons that are all starting with the letter C. So I think one of the most important lessons we learned is the importance of co-localization. That would be bringing the dry lab and wet lab teams in close proximity, having to build a culture and organization that allows for rapid communication is obviously so critical. But in this remote, as kind of increasingly, it seems that their remote is the default for a lot of work within some of these cross-disciplinary teams. There are certainly a lot of benefits to be gained by having, closer communication. And the lessons you learn really in that are how to identify, or how our team members can help identify blockers across teams and how to remove those blockers. So for example, if you're getting caught up on some routine calculations on the wet lab, you might not realize that the workflow improvement or the tools and technologies you need for those simple calculations are just like at your fingertips, but they're always just a little bit too far if you're not right beside someone you're working with. That's co-localization. Co-education is obviously a classic. Cross-team learning, including paper reviews, lunch and learns, pan-science team meetings, as I'm going to call them, like with both teams all in one place, is really a great opportunity to go outside of your comfort zone and get some of that terminology and jargon from one side or the other kind of minimized. It seems like a no-brainer that would be part of it, but it is a trade-off in that you can invest however much energy you want to a point where you're basically teaching lectures or you're teaching a whole lesson plan. You can also back out a bit and sort of take things on in a Q&A format, as long as there's a nice open communication between the teams. We felt it's very important, though, maybe the most critical of all of the C's is the co-ideation. And this is coming up with a mutual understanding of what's possible across a computational and experimental team, because there really is not only that education barrier, but an important understanding needs to be had about what is the optimal data set for both parties. I think the best example of that is like the experimental team asking the machine learning team, how much data do you guys need for your models? And the ML team saying as much data as possible. But obviously that's not practical. So there has to be a middle ground somewhere where you have to pick an experimental approach where you can generate things that are, not just noise or something that's, a quality of data is important just as much as the quantity of data and things like that. So we would have things like datathons where we would actually brainstorm a hypothetical data set where it would be something that we could either make or pay to be made or search from the literature to get some kind of bootstrap. Finally, one of the other C's, one of the ones that's probably pretty intuitive, is collaboration. But why would you want to collaborate across teams? There is such success in big pharma. We're having teams, they do collaborate, but in some sense, teams can be a bit more siloed and be very extremely effective. Cross-functional collaboration is when you have a common objective, does allow you to, of course, with a smaller team, allows you to see more about the problem and what is unique about your problem that you're working on that you can try to solve. The use of the same project management tools in your collaborations is really cool, despite the overhead. For example, in our organization, we have like project management through code, like coding software for software engineers, that we've tried to adopt here at the wet lab and our research side. And some common tools, even just how we plot things. There's different cultural backgrounds for how these scientists work. They could be using wet lab tools like Prism for plotting dose response curves, and it might differ significantly from the culture of data science, where you might be plotting things in Python programming language. But finding when those tools are best to be used and kind of not going all in one camp or the other is just really a good way to encourage all backgrounds to contribute. Moving along now, co-ownership is an interesting one about shared responsibility of deliverables or batches, in our cases of peptides. So signing off on peptide selections, rationale, and hypotheses that underlie the selection of these compounds is something that's you can take for granted a little bit as far as, if you're an ML team member, you can have the best predicted compounds or the most diverse compounds, or when you're predicting these, you can hand them over and sort of say you're done. But really, checking across teams is very important. Having the chemical intuition and the scientists look at the ML predictions, and having the ML team score predictions that may be sourced from structure-based drug design, and kind of getting those two mesh together is really an interesting way, or just really a great way of having people feel invested as a team in the creation of new peptides. Because, together, you really are aiming for the same thing. You have the same goals. You need to celebrate these wins together. There's not really a need for blaming or saying like, oh, this batch of compounds was not as effective as our predictions, or this chemical intuition, it just scored poorly on ML. That would be the retrospective. That's an important part of this process. But revisiting these things on any topics like success of peptide synthesis we talked about, or maybe errors in function prediction are really important. That's that co-ownership. Moving through, I'm almost done. Co-funding, getting the financial support for R&D on methods development for both sides is really critical. There can be a cost asymmetry. I guess the cost of training an ML model, okay, if you're training AlphaFold, that can cost a lot, but more often than not, it's actually relatively cheap to, beyond the man hours of an ML team or a computational team. Most of the expense is, of projects typically revolves around the synthesis of peptides and the functional testing of peptides and those expenses for reagents. But that doesn't mean you can't really treat the whole project as like a lump cost, where you're really trying to perform R&D with a similar goal. And if there's a joint project you can create about creating an ML driven project, or not always validation, experimental validation of ML, but something that's more bidirectional, is kind of the best type of project you can co-fund. And next, I think, is just co-development. Some of these things, I guess, are a little more cultural, but you can be in a position with new technologies like computational methods where they're advancing so rapidly, and the development part of things is so critical because things are changing so quickly. If you were to build a computational platform in machine learning a decade ago, of course, it would look vastly different than what it is today. Similarly, even on the wet lab side, there are new techniques being developed that really greatly expand the capability for high throughput screening of peptides. And you need to be open as an organization, as a team, to basically go outside your comfort zone again and try some of these things, even if it means taking a step back with regard to what your platform can and cannot do. There's kind of like a traditional way of doing, I guess, treating peptides as small molecule drug discovery programs, and then there are more like alternative drug development pathways from the biologics world, like display screening, and weaving your path through all these methods and finding something that's best suited for your team is so critical as you develop your peptide drug discovery team that's cross-disciplinary. So I guess I wanted to just wrap up by saying that there's a lot of lessons. Some of them are very specific to our organization, and some of them, like these ones, these 7 C's, are a bit more broad. And maybe they all seem very intuitive to you, but putting them into action and actually making some of the choices to prioritize some of these things can actually be pretty tricky as you're going throughout your journey through research and this challenging peptide drug discovery game.